It's now time for Q&A session, and I have some questions. Uh, the first one is to Lynn Pure from Gabriele Lombardi. The acceptance highlighted in the UK's public is comparable to the Southern European one? It's a question. So is the acceptance highlighted in the UK public comparable to the Southern European one? And are there studies in Mediterranean areas of these acceptance levels? My feeling is that in Southern Europe, the concern of public about these matters are more present. Lynn.
Okay, so it depends which technology we're looking at. So there's plenty of research about genetically modified foods in the south of Europe, if you like, and certainly some concern. I don't know of any research that's been conducted on synthetic biology and its applications to food production, agriculture, that have specifically considered Southern Europe. The data I showed utilized Chinese consumers. And I think, to be honest, it's a good issue that needs to be looked at.
Okay, thank you. Next question to Neil Stevens from Joanna Prata. Considering the challenges of cell cultures with microorganism contamination and antibiotic use, as well as other resources use, what is done to control the presence of antibiotic residues in culture meat? And what is the environmental impact of culture meat? Are there life cycle assessments available comparing, for instance, with vegetable protein alternatives? Thank you.
Great, two really great questions. I'll start with the question about contamination, but I'll put in the caveat that I'm, of course, the sociologist exploring the politics of all of this. So my technical answers might be different to the people that are building the bioreactors and the rest of it. But yeah, so contamination is a real issue, obviously. You mentioned the possibility of using antibiotics to stop forms of contamination in the bioreactor systems. Overwhelmingly, the people involved in this say they are not going to go the antibiotic route because they believe, first of all, they believe they don't have to. I know that there are people that question whether it will be feasible at scale without antibiotics, but yeah, I spend quite a lot of time talking to people in the community, and they seem, I mean, they always seem confident about everything because it's their job to seem confident about everything, but they genuinely seem really quite confident. I believe them that they seem absolutely confident that antibiotics is not going to be necessary, and sterility is maintained through very tight control of the system and batch destruction. Basically, if you got a bioreactor that gets contaminated, you just destroy the whole lot. You have a procedure for doing that safely, and of course there's an economic loss with doing that. Scare is much more expensive than throwing away a bioreactor's worth of material. So it's generally through having very high quality sterile environment, having really high quality quality assurance procedures to try to prevent contamination getting in, but then batch destruction essentially. How successful that will be, how costly it will be, these are open questions, and they're questions that I think, as much as I think the community are confident that they can address that, I think it's questions that we still need to be asking because they're vital. The second question, part of the question, was about the environmental impact. There absolutely are life cycle analyses out there. They have different findings generally. One of the things that you find is that it does depend a bit on what you compare it to. So you mentioned plant-based foods. So I don't think there's any, I mean, maybe say there isn't one is a bit strict, because there's quite a few of these life cycle analyses now. But none of them really say that cultured meat is less energy intensive than producing, say, chickpeas. You know, it depends what you compare it to. It's normally best compared to beef, because beef has a higher environmental impact than, say, chicken. If you just look at, I'm not talking cultured now, if you just look at livestock-based meat systems, you know, chickens have a much lower environmental impact because they have very short lives. They're much smaller, right? So, you know, whether cultured meat is more efficient than chicken, different studies come out with different responses. Equally, I want to stress the extent to which all these life cycle analyses are really speculative, because they're trying to model a system that does not exist. They're full of hypothetical numbers. And it's not even that they don't know what the precise numbers for each variable will be. We don't really know what the variables themselves will be, because you don't know exactly what production method you're going to use. And to go back to something we had from the previous talk, it also depends a bit on whether you want to use a system that uses gene editing or not, because you can gene edit cells to make them grow more efficiently. But if you do that, you then potentially risk another reason for people to not like your cultured meat. So some companies are using those approaches and some aren't. So unfortunately, my answer there is we don't know, but I think it's important that we recognize that we don't know. I think that's an important insight. Thank you.
Thank you. One more to Ms. Stevens. From Joanna Prata again, are the cell lines used in culture continuous or mortal, or they need to be collected regularly from animal donors? How does this affect public acceptance?
Another fantastic question, another at least two-part question. So you can use both of those types of cell lines. You can use immortal ones. You can use something called induced pluripotency system to make a cell line. So this is one of the ways that you sort of modify the cells. So what we're talking about here, for people who aren't following the question, is whether you can get a cell line that will kind of infinitely produce cells that you work with, and you never need to go back to the animal, or whether you have a system where you go to the animal, you know, once every a period of time, whatever it is. It depends on the, you know, how much stuff you're making, but whether you need to go back to the animal and a cell line will only work for 6 months or 2 weeks or 2 years or whatever it is. It's unlikely to be 2 years if it's one of those. But anyway, that some people have looked for. So typically a cell line won't be immortal unless you modify it in some ways, but you can sometimes find kind of mutations that give them that state that are just kind of found in nature. And if you can get those cell lines, then you can have an immortal cell line. Whether they really are immortal and whether they really do last forever is a different question. But absolutely, different companies are using different approaches, and it's a little bit related to the previous point about what kind of modification of the cells you want to do. So some of the companies that want to minimize the amount of sort of scientific intervention are more likely to go to the animals more regularly, so you don't have to modify the cells, and you don't get the GM phrase or the gene editing phrase sort of just in the same kind of waters as you. So public acceptance, I don't think the surveys have got to that level of detail, and I think there's lots of reasons why I critique the surveys that have been done. I think what we heard in the previous talk about issues like the deficit model, I think that frames the thinking of a lot of the people that do studies of publics. They're very much framed as consumer acceptance studies, which generally takes all of the complexity that people have as cultural figures with political aspirations and reduces that to a yes/no purchase decision. Will you buy it, yes or no? And I don't think that's good enough to understand the complex politics of something like this. But those studies don't really get to the level of detail of looking at cell source like that. And there's an extent to which I'm not sure they need to all do that at this point, because I don't know if that will be on the labeling unless it invokes sort of genetic modification regulation. So brilliant question, but a question for the future.
Thank you. Now question to Lynn from Rostislav Bubnov. What instruments do you consider effective to decrease culture-associated negative emotions in population? We can't hear you.
Yes, you
Yeah.
can. There you go.
Very good.
It's an interesting question. I have to ask why you want to do that. Why would you want to reduce negative effect?
I see. Very good question back.
Sorry, could you clarify?
Yeah. And so the question was, what instrument do you consider effective to decrease culture-associated negative emotions in population? Well, I suppose that these attendee would 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 like to understand how to change the perception of consumer that that is associated to the culture, the background they have. That the way I understand the question.
Okay, so thank you very much for the question. It's an interesting one. I suppose I would not want to use instruments to do that. If people have a negative emotional response to cultured meat or synthetic biology, then that's the reaction they have. And I think that's potentially a very legitimate reaction in the same way as having a very positive reaction, a very positive effect might be. So I wouldn't want to change that. I would like to look at it in that cultural context and understand why it's arising. But I think it speaks to Stephen's question and the kind of talk that he's given. You know, we cannot expect people to automatically consume the products of technology. There'll be a big spread of reactions. They may not be homogenous. And we might also want to consider that sometimes people are thinking as citizens. A vegetarian can have a view about how chickens are raised for meat without consuming the meat of chickens. And they would have a negative affective response and potentially campaign or read or have views of regulatory frameworks in line with those responses. So it's not really answering your question, I'm sorry, but I mean, that would be my perspective.
And Neil, would you like to add something to this?
Um, so I mean, my work as a sociologist is not, there are people that do this, but I don't do sort of focus group studies with publics. My own work isn't directly looking at public perceptions. I interview the group, the scientists, the entrepreneurs, who are looking to produce the technology. But that given, I think I would broadly agree with the previous answer, that recognize that you know, I think that as analysts we need to grapple with the complex cultural emotional components of all of this, not as something to be sort of controlled for, if I understood the question, and maybe I didn't, but something to, you know, you need keeping the messiness of your social science data. That's where I would be.
Okay, thank you very much. Okay, so thanks again for your presentation. Thank you very much.